So now let's look at each of these stages a little bit more closely. We won't focus on the images stage where we're getting that activation pattern in the back of our eye. We'll go to the next stage and start with the primal sketch. Now, Mark tells us the primal sketch is a viewer-centered two-dimensional coordinate system. It includes information like zero crossing blobs, terminations, and discontinuities, edge segments, virtual lines, groups, curvilinear organization of boundaries. I sort of think of this as a kind of Toulouse-Lautrec of the image, right? Just line drawing, essentially, figuring out the general outline of the image by trying to determine the overall lines and organization there. Next is our 2.5D sketch. Mar tells us that the 2.5D sketch is a viewer-centered 3D coordinate system that contains local surface orientation, which he represents using needles as primitives, distance from viewer, discontinuities in depth, and discontinuities in surface orientation. And here's a picture from a paper by Mar and Nishihara on representation, recognition of spatial organization of three-dimensional shapes, in which he talks about that. So we have a two-dimensional depiction of, I don't know, like a birthday cake, essentially. And then all these little needles on that indicate surface orientation and relative distance from the viewer. Here's a different approach to the same kind of thing, right? This is from a lab in UCLA. They take this original picture here, and then they compute a 2.5D sketch through these two stages, right? Where we get three different surfaces, their relative position given the viewer. And finally, we get to the 3D model. The 3D model is an object-centered 3D coordinate system. 3D models arrange hierarchically, each one based on spatial configuration of a few sticks or axes to which volumetric or surface shape primitives are attached. And what does that mean? Well, essentially, it's objects. All right? So, for example, this again from that Mar and Nishihara paper, we have a 3D model of a human being as a series of cylinders that are volumetric shapes that are around these stick axes. So we have the head axis, the torso axis, the legs and arms, and then we have a hierarchy of models there. If we look at the arm, we have that breakdown. If we look at the forearm, we have this breakdown. When we look down here at the second half of it, we get the breakdown of the hand. So we have this hierarchy of shape models from the original overall model. Here's a different way of thinking about that. So we have this depiction here of a pear and a potato, and our first pass then creates these cylindrical orientation volumes, and then we fill in our surface shapes, and we start to get images of objects in their relative position, and we can even rotate them around in this particular computer-generated reconstruction. So this is the idea that Mar has. We start from these reflections of light intensity at the back of our eyes, and we're trying to generate this hierarchy of representations of shapes and their relative positions with one another. So Mar definitely kick-started visual research and influenced the way that computational explanations were approached within cognitive science. But Mar also mischaracterized vision in important ways. And ironically, even though he was refining our sort of proto-theoretical notion of vision and trying to refine our way of explaining that using computational theories, he still includes things in it that are sort of our ordinary intuitive notion about what vision really is and how it functions. And we'll start with this first one, this first way that he mischaracterizes vision in this lecture. We'll finish the lecture with that, and then we'll move on in the next lectures as we go through and start to explain visual processing, for instance, in the primary visual system. we'll start to see the other ways in which this view breaks apart. So the first problem is that MAR has this view that ultimately we're working through a series of operations, computational operations, to create a single general purpose representation of objects and their positions in the world. However, as vision researchers started to refine and work on their understanding of vision, they started to realize that vision seems to contribute to 3 different sorts of spatial maps with different resolutions and even different origins. And so we'll look at that to end today's lecture. As we start to look at how primary visual system works and higher order visual processing works, we'll start to see that vision isn't a single stepwise processing stream. The goal is to generate that single representation. There are different maps. There are also different processing streams. And vision is actually an integration of a number of different processing streams, as we'll see. So on one hand, right, one of the first dissociations that people discovered was a dissociation between visual processing for motion perception and motion generation or motion guiding. And this is often referred to as the how-where pathway that goes upward to the partial parietal lobe. The other one that was distinguished is fine pattern or object recognition, sometimes called the what pathway, that goes down to the inferior temporal lobe, as we'll see. And these pathways originally were thought to only get dissociated after they reach the visual cortex. But then research started to indicate that that processing of different streams for different purposes began all the way back in the retina. And as we'll see, that means that vision isn't a single serial processing event. The next thing that kind of leaped into visual processing research was the realization that while we have this very stable, seemingly highly detailed perception of the visual scene, right? We seem to see the whole visual scene. It's very stable. There are lots of details. In fact, that's not true. In fact, it's not true because it's computationally untractable for us. Even though a tremendous amount of our brains are devoted to visual processing, our brains would have to be, I don't know, the size of refrigerators to actually process all the visual information that we have with anything like the detail we think we do. And so we saw this phenomenon with change blindness, inattentive blindness, and so on. Right? A huge part of visual processing involves maximizing or optimizing our resources. So that small area, that foveal area that has all those cone cells, right, that's really geared towards fine-grained, color-sensitive object recognition sort of stuff, that little bit of resource there, that takes up a huge amount of processing. And in order to be able to get that kind of information, we can only use that little area of the visual field at any given moment. And so we foveate around. We move that area around to get information from other parts of the visual scene to integrate it into our perception. But in order to do that, we have to have a mechanism that picks out what we should integrate, what we should be looking at, and that also keeps the whole visual scene stable across all these rapid movements of our eyes, like 3 times a second. And so we'll see that, in fact, there are other processes that pick out things for us to attend to, and processes that create a stable visual image for us as well. So we'll look at these different spatial maps in our next module.